So just wanted to start by thanking the organizers for the opportunity to share this story. I feel like, the world was watching as COVID hit New York City. I was actually there with my 70-year-old plus parents over the weekend of March 6th through 9th for a kind of bucket list trip there from Canada, and they had never been to New York City. So I brought them to 2 Broadway shows, to 3 museums. We went to the Chelsea Market, and we were just right in the thick of it, just as we were beginning to appreciate the impact that COVID is going to have on the city, and on the train, of course, on Boston as well. And so this is a story really about how the outbreak unfolded in the Boston area and in Massachusetts, but I think it also provides some generalizable insights about the dynamics of importation and transmission and the impact of superspreading that can be lessons for other cities and for the world as we try to control this bug in this next wave. So just, oh dear, how do I advance? Oh, there we go, okay. So as with, I think, virtually any work on COVID, this has been just a massive collaboration between my colleagues at the Broad, and working really in very close collaboration with some of the hospitals in the Boston area, in particular with Mass General and Brigham and the Partners Network, and the state public health department and public health lab. So we're closely trying to use genomics and this genomic data both to understand the virology, the genomic evolution of SARS-CoV-2 itself, but also to use that information to understand and hopefully inform outbreak response. And we're also working closely with the CDC, with our epidemiologist colleagues in Harvard School of Public Health, UMass Medical School, and Rhode Island. And as time goes on, this collaboration is really growing. And so I think it's fair to say that that COVID-19 is really a watershed experience. I mean, for so many reasons, but in my little corner of this, for the application of genomic data to understanding and controlling, or trying to control the spread of the virus. And this just goes to show that this is actually just a screenshot of the global SARS-CoV-2 sequences in the next strain platform just from this morning, where you can see that sequencing of viruses from infections is happening really around the world, and our picture of how the virus is evolving and spreading and how we can use this information is growing really by the day. And so, science and genome junkies and epidemiologists are using this information in new ways, but I think it's also kind of permeating into the public domain and public consciousness. And I think for the first time ever, my friends and family know what it is I do now and are starting to get some appreciation of how it might be useful. And this was just a great piece that I wanted to flag in the post back in October about how we can be using genomic data more powerfully, and some of the missteps and corrections the US is doing to bring this approach online at scale. And I would just suggest anyone have a look at, they have a fun infographic that I think represents it really well. But, and I just also wanted to mention, just by kind of context, the work that we do. So I'm based at the Broad and the School of Public Health, Harvard School of Public Health, working closely with colleagues including Pardis Sabeti and Diane Wirth and some others who have a long history of bringing kind of genomic data to the problems of infectious diseases, mostly in the developing world, in particular with a focus in West Africa, where we've been working for over a decade to establish genomic capacity for tracking the spread of infectious diseases, for responding to outbreaks, for tracking the spread of drug resistance and other kind of molecularly relevant signals. And in many ways, the work that we did in the Boston area during COVID has been standing on the shoulders of the learning that we've done together with our colleagues there in these much more challenging environments where infectious disease threats are a constant. And so it's been a great opportunity to bring that, that experience to bear at home. And this is just a couple of examples where in particular during the Ebola outbreak in 2014 in West Africa that really impacted our colleagues there. We were able to use the data to identify that transmission was human to human. And in that case, it was originally thought to be repeated introduction from the environment, and just the parallel there of trying to understand, how transmission is happening with COVID. And then using genomic data where we can to actually infer transmission links, where epidemiology data and contact tracing kind of falls down given resource constraints and complexity. So it's very much paralleled. But of course, we're interested in what is happening with COVID, and just a quick overview of where we are, where we've come from in Massachusetts. So the first case that was reported in the state was on January 29th. That was a traveler returning from Wuhan, in fact, but that case was identified and quarantined, and there was no onward spread of that we've been able to see either epidemiologically or in our genomic data. But things really exploded early in March, in particular, kind of burst onto the public awareness following an outbreak and apparent super spreading event at a biotech conference that happened just at the very end of February in Boston. And from there, things escalated. The first wave peaked in April. And of course, as in many areas, including New York City, the transmission in particular affected congregate living settings and other high-risk communities. And of course, we're now, I'd say well into what I think is safe to say is the second wave at this point, with 226,000 cases to date and almost 11,000 deaths. And I just did a quick back of the envelope calculation, and that seems to be track on par with numbers that we're seeing in the state of New York, just if you adjust for population. So we are right there with you. And so as the epidemic kind of unfolded or really began in the Boston area, we on one hand, we're working hard to make sure that our colleagues in our partner sites in West Africa were protected. We sent PPE, we sent all the reagents to do qPCR diagnostics at large scale there. We were working with them constantly, but also focusing in our own city, in our own backyard, and in the state to ask these basic questions of how was the virus entering the state, what was driving spread. We didn't really understand or anticipate at that point just the dramatic impact of clusters and super spreading events. And then really in a real-time way using the data to answer questions that were relevant to public health, to the hospitals, and hopefully help them inform their response strategies. So just very briefly, a few words about the approaches that we use for sequencing. I'm not going to go into detail here, but just to say that we have two strategies that we use in the lab to capture the viral profile of samples that are collected from infected patients. Mostly we're working from residual diagnostic materials, so sample that's left over after a diagnostic test is run gets diverted to our lab, which is basically just an academic lab at the Broad Institute. The sequencing that I'm talking about here is being done by RAs and postdocs, students, not by the big kind of Broad testing machine that you may have heard of. It's really kind of an academic pursuit done at academic scale, just working around the clock on it. We use both unbiased metagenomic sequencing, where we do not enrich for SARS-CoV-2 prior to sequencing, and that gives us a picture of other respiratory pathogens that may be in the sample. It's a bit slower, it's a bit more expensive, it's a bit more laborious data to work with, but it does give us that unbiased picture of the entire kind of profile of the sample and an unbiased picture of the kind of evolutionary state of SARS-CoV-2 in the sample. But then, by contrast, we also use a targeted approach, an amplicon sequencing approach that just allows us to do really quick turnaround on the order of a couple of days of sequencing, where we pull the SARS-CoV-2 out of the sample and sequence that specifically. And the graph here just shows that the performance of these two approaches as a kind of function of the viral load, or the CT of the sample. So the sensitivity for metagenomic approaches kind of falls off around CT 30, which is still pretty high, pretty sensitive. And for the Arctic or amplicon sequencing approach, we can get very high CT or very low viral load genomes recovered up to about CT 35. And this is just an overview of that process, mostly to highlight some of the folks in the lab who really were literally working around the clock on this, and coming in. Most of the data I'm going to show you is from that first wave between early March and mid-May. And while many of us, including myself, were safely working from home, these folks were coming in every day and handling virus in the lab to get this work done. And then also, very quickly, I'm not going to go into details of our analytical approach, how we kind of handle, clean, and analyze the data. All of our methods are entirely in the public domain. I'll speak to that in a moment, but I'm happy to answer any questions. But just to say, largely it's based on alignment to the Wuhan reference and calling consensus SNPs. We use other approaches in some specific applications, in particular going deep on inter-host variation, which I'm going to get into. Is a very active area of research in the lab, in particular looking within host tissue tropism that's relevant from the last talk, and looking at autopsies and looking for the occurrence of reinfections or recrudescent infections in some other work that is beyond the scope here, but very active in the lab. And here, this is just the environment where, if you're interested, all of the workflows, all of the data are publicly available in this Terra workspace, which is a platform for data analysis and data sharing that the Broad supports, and happy to point you at that if you're interested. So now, just looking at the data set that I'm going to be describing, and kind of orienting you within the state. So the data set focuses on about 800 high quality, 98% complete SARS-CoV-2 genomes from the first wave, and the data set is biased towards Metro Boston. That's partly because that's where most people are. That's also where most COVID was. And that is, largely the catchment of the hospitals that we're working with as well. So we are biased to the Boston area, but have some sense of what was going on across the state at the same time. And just wanted to highlight these particular communities just north of Boston: Chelsea, Everett, and Revere, which were the hardest hit. These are cities, just outside Boston city itself, that saw the most cases. And are highly represented in our data set. And the other thing to note here in this panel is just that we were lucky, I guess, to be able to recover a lot of the cases that came through initially. So even though it's over, we track pretty much one-to-one over the first 2 weeks or so, and then it diverges quickly and widely. And now at this point, I think we've sequenced about 4% of the total cases in Boston. But this early representation just turned out to be incredibly important, to be able to capture COVID as it was coming into the city and into the state, and then watching it spread from there. I think it's really added a lot of power and resolution to the data set. So now this is, I'm going to be showing you quite a few of these phylogenetic trees. I'm guessing you probably have some familiarity with them, but few are true experts in really unpacking what's going on here. So I'll try to walk you through, or feel free to ask questions. So you're looking at here a time tree of SARS-CoV-2 with where each branch, or each tip, sorry, is a is an individual genome, and they're colored by the origin of where the sample came from. And so our data set is in red, and it's against a backdrop of SARS-CoV-2 from around the world, subsampled from the GISAID database. And so that just puts our data in the kind of global context. And so what you can see just at this kind of very macro level is that first of all, there were many introductions into the state. You can see there's red lines here kind of sprinkled throughout the global tree. But that most of the introductions, at least in this representation, have come via Europe, which I think now is well known, but early on that was just a really important observation, both in terms of understanding how these introductions were happening, and also for kind of rebuting the political narrative around the role of Asian countries and China in particular in driving transmission domestically, and this is true certainly in Massachusetts and in New York, and quite widely across the country, as we now know. We can see that there were many introductions, in particular early on. Depending on the model, the numbers hover around 100 to 120 introductions of distinct viruses early on, within the first few weeks of the data set and of the epidemic in Massachusetts. This dropped over time, and by April already there were relatively few new introductions, at least that we could discern genetically, which is a point that I'll just come back to in a moment. But even in this data set, in May, we start to see an uptick in introductions, and this has continued over the summer. So with relaxing of travel restrictions and stay at home, and just kind of relaxing, I think in our minds to some degree, we're seeing more introductions into the state. And just a quick note now, I think this graph is really unsatisfying. I wish that I had the forethought to do a better job of representing this to you all, but there's a story here that we haven't been able to fully unpack yet, but that we're actively working on, that is the kind of recurrent introduction and recurrent connection in the Northeast. And I think given the kind of population centers between your city and ours, the connection really between New York and Boston, and other cities and communities as well. But in addition to the roughly 100, 120 discrete introductions into Boston, we think that there's been there was a tremendous amount of kind of recycling and sharing between our cities and between the cities in our region. That was driven, in part because of the lack of interventions, which, very hard decisions to make, but not enforcing border closures or quarantine for travel between our states undoubtedly led to shared transmission across state lines. It's obvious, but we're able to see it, and we're getting to the point of quantifying that now. And so in terms of what we really saw, and kind of now starting to zoom in on the dynamics at a higher resolution within the data set, what became very clear was that, what became very clear was that most of the introductions were singletons. So many of the introductions that you see here did not lead to onward cases, but much of the transmission was driven by just a few introductions. And this has been connected to superspreading events and to other clusters, and has been seen elsewhere, but is really represented in our data. And so this, in terms of the kind of genomic epidemiology, is a concept of overdispersion, and the dramatic overdispersion that we're seeing with respect to COVID. And this is just based on the idea that most introduction or most transmission events are discrete and independent. and most lead to only, very few onward infections. But the distribution of transmission events for COVID, we've seen here and elsewhere, is highly overdispersed, meaning that we have many examples where there is a disproportionate number of onward cases from a single transmission event. And some of the most kind of extreme of these are superspreading events, where a single individual infection results in many onward infections. And this is defined in different ways and used colloquially kind of loosely, but we use a definition of at least 8 onward infections from a single individual. And so this is just reiterating the idea that most cases do not result in downstream infection, but some result in many. And although superspreading events are rare, they really result in a disproportionate amount of infection and disease burden. And so I'm just going to draw your attention now. This is another representation of the tree, focusing on a few of these examples. So just quickly, so I'm pointing here to this cluster at the top of the tree, which I'll explain in a moment. You can see there's just different patterns of introductions and onward spread in the tree. So this example with the arrow, you can see it's basically one introduction that led to many cases, but did not go a lot further. And then you can see here an example where there's an introduction that led to many initial cases, but then also many onward cases and community transmission, which I'll describe. And then, as I mentioned before, you can see many examples of introductions where there's really no onward transmission. And so focusing here at this first cluster and superspreading event was in a skilled nursing facility that is associated with MGH Mass General Hospital, where they actually tested initially a subset of the residents in a prospective way. They didn't anticipate that there was COVID in the facility when they initially did testing. They were testing because they're planning to relocate residents to make room for COVID patients. But they found in that initial testing that 60% of residents were asymptomatic but positive. And so that incited extensive testing of all residents and staff. And over the coming days, it turned out that the vast majority of both residents and staff were in fact positive, and many were starting to develop symptoms at that point. And so we were called in really to sequence these genomes to understand how this transmission was happening and to understand the relatedness between these infections. And what we saw was striking and really just a great microcosm of this idea that some introductions go nowhere or have very little onward consequence, whereas others really can be devastating. And this, and so what we saw was that there were in fact 3 introductions represented in these network diagrams into the facility. 2 of the introductions led to 2 onward cases, but 1 introduction led to infection in 78 individuals over a very short period of time. It looks like it happened over the course of a couple of days, just by the fact that there was so little genetic diversity in the virus, and that the timeline of the testing. So it was just a really striking example of the circumstances under which a single introduction can really have devastating consequences under the right conditions, whereas others, for reasons that we don't fully understand yet, did not have the same impact.
Okay, Bronwyn, roughly a 2-minute warning.
Oh, 2 minutes. Okay, great. So I'll move on. The next example is International Business Conference, which has attracted a lot of attention, where a single introduction that was marked by a genetic variant that we were able to track led to ultimately about 30% of cases in all of Massachusetts. And just in the Boston area alone, within our data set, led to about 20,000 cases. this happened very early in the outbreak. As I mentioned, this was in late February, and the circumstances were just ripe for kind of a perfect storm of unfortunate events that led to the transmission widely across the city before we were really aware that it was present, or that the measures that we've now become used to were needed to prevent the spread. And it spread not only within the Boston area, but also widely domestically. There were cases carrying this particular marker in 29 US states, and led to community transmission in at least 13 of those, as we can see in publicly available data. And within our data, we can see that that viruses from this super spreading event led to extensive, as I mentioned, transmission within the city. And in particular, this is just looking at the tree another way. So the conference-associated samples are yellow here, and in red you can see spread within the Boston area homeless community. Where there were at least 7 introductions in our data set, and many of those infections were a result of descended viruses from the super spreading event. And so I think it's just a remarkable illustration of how a single super spreading event can lead to just such dramatic onward consequences over time. And the transmission continues. I think I can just skip. This is just showing how viruses from the conference seeded infections in these high risk, highly impacted communities of Chelsea, Everett, and Revere, and in homeless populations, and just spread really quickly through the city. By even the middle of March, we could already see transmission of conference-associated viruses really throughout the city in space and throughout socioeconomic demographics. And the spread of this virus continues within the city, within the state, across the country, and really around the world. There are in fact two marker mutations that continue to spread, and we're tracking those just to really understand how viruses move throughout populations. And I think I can. Well, I'll just quickly say that we're tracking other mutations. These are just benign markers. They have no functional effect to understand and model how viruses are moving through communities. And so that is ongoing work, but really fascinating to be able to see at this level of resolution what are the sources and sinks of infections, and how viruses are moving through the community. And then finally, just to say, not all apparent clusters are clusters, in fact, or are super spreading events. And a lot of our work has focused on investigating relatively small clusters, apparent outbreaks in specific communities, in hospitals, in schools, and workplaces, where you're vigilantly using infection control measures, but of course the virus can escape through. And the question, when you get a case or 2 or 3 or 4 at your school or workplace or hospital, is, do we have an outbreak on site? Do we need to close the campus? Do we need to decontaminate the ward? Do we need to do something different? Or is this just introduction from community spread? And this just shows that early on, and in work that we're doing very actively in a continuous way, these are 2 examples where apparent clusters at Mass General Hospital, we were able to show with genetic data, show no evidence of being associated. They seem to have been introduced independently from the community. And that was just really reassuring for infection control at the hospital, to know that their infection control practices seem to be working, and that these cases are unassociated and independent. That's just a quick comment on metagenomics. The upshot is that we haven't found a lot of co-infections, but we'll be looking through the spring and summer, but we'll be looking at this now as we enter the flu season. And that's just to reiterate the key points, which I think I can skip, and ongoing work, and just to acknowledge the amazing crew that's been involved in doing this work and continues to work hard on these problems. And here are just some of the resources that we share in our socially distanced group photo.
All right, great, Bronwyn. Thank you very much. Very cool. Obviously, knowing about these is giving us some hope of trying to interrupt those events.
Right.
But obviously, knowing that they happen is the most important thing to start with.
Yeah, I think that one of the takeaways from this work is that, you know, the contact tracing and public health investigation of clusters. I mean, the estimate for the conference was 100 known conference-associated infections, and now with genomic data, we can see the true impact of these goes far, far beyond that. So it's just a great reminder to us all. You know, at that point, I think we collectively didn't really know better, but now we do, and so I think it's a great reminder that the knock-on effects can be devastating.
So we should be quick, but so we have two questions that are maybe the same question from Andy Marks and Rob Schwartz. The question roughly is, do we think the super spreader events are viral variants that made them more transmissible, or is it something else in the environment?
Yeah, I think it's pretty clear that there is nothing special about these viruses. That the you know, the circumstances that drive the spread is really based on human activity one way or another.
And a related question that Andy has is, are the super spreader events a single genome typically, or more than one? I think the data would say one, I guess.
Yeah, so there's different circumstances. In the nursing facility, it was a single genetic genotype that drove the vast majority of spread. At the conference, there were at least two, but on the second one was on the first background, so it wasn't like there were two super spreading events, but there was evolution. We think at the conference that kind of defined a sublineage of the first that was associated with the same kind of contextual super spreading event that led to the onward spread. If that makes sense.